there are other types. So before I go into the other types, so I'm going to go over how they do thin layer and paper chromatography. So like I said, after the plate has the sample spotted on the stationary phase, it's placed upright in a developing chamber with a small, shallow pool of solvent, also known as elutant. So as the absorbent is in the developing chamber, the solvent will slowly move up the TLC plate or the paper plate by capillary action. So as the stationary phase absorbs the mobile phase will carry the different compounds, and they travel at different rates based on the properties of the compound, the mobile phase, and the stationary phase. So the mobile phase is usually in an organic solvent. So this, because the mobile phase is an organic solvent that's nonpolar, the nonpolar compounds will travel farther and faster during the chromatography, because the nonpolar compounds will not adhere to the stationary phase, and since it's like the organic solvent, it will travel with the organic solvent up the plate as the chromatography happens. So the polar substances will not stick. It will not travel as much with the organic nonpolar solvent because of the differences in polarity. The polar substance will stick to the stationary phase more and will move less during the chromatography. So there's another type of chromatography known as column chromatography. So this is when you have a column filled with either silica or aluminum beads, and that's used as an absorbent for greater separation. So this is similar to like TLC chromatography, except it's on a larger scale, and you're able to separate and distinguish larger quantities of the sample. So in this type of chromatography, use gravity to move the solvent and the compound down the column. So as you can see on the right, you have a column filled with sand and a glass wool at the bottom to filter the compound, to prevent any of like the alumina or silica from getting into your collection flask. But then you have the sand and the alumina to be used as the stationary phase during this chromatography. So when you pour the solvent down the top of the column, the alumina and the silica will slowly separate the solvent into its different compounds. So the compounds that are more like the solvent will move along with the solvent down the column faster and farther, while the compounds are more like the alumina or the silica. It will stick to the alumina and the silica more, and will move slower, and will not move as far. So this causes each fraction. So it will cause each compound to leave the column at a different rate, and these different compounds that drip out at different periods of time and at different rates are known as fractions. So each fraction will contain a different compound that travel through the column at a different rate. And once you collect it in the collection flask, you can evaporate the solvent in order to leave only the desired compound behind. So that's column chromatography. Next, there's another type called ion exchange chromatography. So this is when you separate the compounds based on their ionic charge. So the beads in the column instead of like silica or alumina, it will contain beads in the column that are coated with charged substances to attract and bind molecules with the opposite charge. So if you have a column full of ionic substances, it will attract the compound with the opposite charge. So if you have positively charged substances like cations, it will attract the negatively charged compounds and will stick to it and cause it to travel down the column at a slower rate. But if you have a column full of negatively charged molecules, anionic molecules, the positively charged compounds will stick to the column more and travel slower and a shorter distance. So the stationary phase is composed of a charged ion, whether positive or negative, depending on which compound of the mixture you want to separate out. So if you want to collect the positive charged compounds, you will have a column full of negative charged molecules in order to have the negatively charged molecules adhere to the positive compounds and keep it in the column, and the opposite for the opposite charge. So you can change the stationary phase based on which compound you want to collect in the column. So like I said before, anion exchange, this is when you have the stationary phase composed of positive ions that will attract negatively charged substances and keep it in the column for you to collect. So everything else will pass through, but the negatively charged substances will be stuck in the column, and you can separate it from the column later. So anion exchange is when you want to collect the negatively charged substances. This is not named based on the charge of the stationary phase. So cation exchange, this is when you want to collect the cations, the positively charged molecules. So in order to collect that, you would want to create a stationary phase full of negatively charged ions. So you can have them stick to the cations and keep it in the column. So the attracted substances, like I said, will travel in the column more slowly, and the other molecules that you don't want will travel faster through the column, and you can collect that and then put it aside. And then at the end, once everything leaves the column that you don't want, you can extract the substances stuck to the column that you are interested in. So after all the compounds move through the column, you can use a salt gradient to elute the charged molecules stuck in the columns. So you can use that to collect the desired compound stuck in the column. That's ion exchange chromatography. Lastly, there's another type called size exclusion chromatography. So this is when you have a column full of beads, and these beads have tiny pores of varying sizes. So the pores will allow smaller compounds to enter the pores and slow them down. So the smaller compounds will be stuck in the pores of the beads and will travel slower through the column. While the larger compounds that can't fit in the pores will travel faster by moving around the beads. So the varying sizes of the pores will allow the molecules of different molecular weights to be fractionated. So the larger compounds will come out of the column first, while the smaller compounds will be stuck in the pores and will travel slower through the column. So this chromatography separates the compounds based on their size. So the largest compounds will come out first, and as time passes, the smaller compounds will come out until the last compounds that come out will most likely be mostly be the smallest compounds that were stuck in the beads for a long time. So that's size exclusion chromatography. So lastly, affinity chromatography. So this is when you use a very specific substance as the stationary phase. So because you have a very specific substance as the stationary phase, it can attract that very specific substance that binds to the stationary phase. So this is the classic example is when you use a receptor or an antigen for a specific protein, or a receptor for that specific protein. So what happens is that protein of interest will be bound to the substances in the column, because you create a column with a high affinity for that protein. So affinity chromatography is very specific. Only that very specific substance will be stuck in the column, while everything else will pass through pretty quickly. So like I said before, the beads, you can have beads that are coated with receptors that bind to that specific protein, or use an antibody that binds that specific protein in order to retain that desired protein in the column. So you can see on the picture on the right, the yellow is the beads that you use in the column, and since they have specific receptors or antibodies, the targeted protein will be stuck in the column, bonded to the beads, while everything else that don't have that specific affinity for the beads in the column, they'll travel through the column pretty fast and won't be stuck in the column.